chase bacteriophage experiment. This is the early experiment, and we will discuss this experiment in this lecture, tobacco mosaic virus experiment, the structure of the DNA inside the nucleosome, the Watson and Crick model of DNA, the double helix, organization of DNA or RNA in the chromosome. What is the nature of the genetic material? It's a question scientists try to answer. It's a stable source of information. It has ability to replicate accurately. It can be changed. It's stable, replicate, it change. There is a timeline for events. In 1890, Weizmann substance, the nucleic, the genetic material, a substance in a cell nuclear control development. Weizmann. Genetic material is a substance in the cell nuclei control development. In 1900 chromosome shown to contain hereditary information, later shown to be composed of protein and nucleic acid. In 1928, Griffith transformation experiment. In 1944, Avery transformation experiment. In 1953, Hershey Chase bacteriophage experiment. In 1953, Watson and Crick proposed double helix model of DNA. In 1956, Gerer and his colleague demonstrate RNA is a viral genetic material in tobacco mosaic virus. This is the experiment of Frederick Griffith. Transformation principle demonstrate with Streptococcus pneumoniae. Streptococcus pneumoniae is a bacteria, a pathogenic bacteria. Here, Frederick Griffith transformation experiment conducted in 1928 was a series of experiments on Streptococcus pneumoniae that lead to the discovery of the bacterial transformation, a process by which bacteria take up foreign genetic material. In this experiment, Griffith used two strains of S. pneumoniae, a virulent strain that caused pneumonia and a non-virulent strain that didn't cause pneumonia. In one experiment, he injected a mice with S strain, which caused death. In another experiment, he injected mice with the R strain, the rough strain, which didn't cause death. In the third experiment, he killed the S strain, making it non-viable, and injected it in mice, which didn't cause death. And he killed the non-virulent, survived, no bacteria recovered. Here, he combined both the killed non-virulent and the living non-virulent, and inject the mice, and the mice died, and the virulent bacteria recovered. Here the Oswald Avery transformation experiment, and determine DNA is a genetic material. Oswald and Avery transformation experiment in 1944 demonstrated that DNA was a genetic material responsible for the transformation of non-virulent bacteria into virulent bacteria. Prior to this experiment, it was believed that protein were the genetic material. Avery, along with his colleague Colin MacLeod and McCarthy, purified various cellular components of the Streptococcus pneumoniae and tested their ability to transform non-virulent bacteria into virulent bacteria. Here the series. Treat with the RNAs, add DNA to our bacteria, plate growth, S transformant produced, mixture of DNA and RNA. Here a mixture of DNA RNA. We treat it with DNAs, add RNA to our bacteria, plate on growth media. No S transformant appear. Here is transformant produced. Hershey Chase bacteriophage experiment in 1953. The bacteriophage, as we know, a virus that attack bacteria and replicate by invading a living cell and using the cell molecular machinery, hacking the molecular machinery of the cell. Hershey and the Chase experiment conducted in 1952 by Alfred Hershey and Martha Chase was a series of experiments that helped to confirm that DNA is the genetic material in cells. The researcher used bacteriophage, which are viruses that infect the bacteria. To investigate how genetic material is transmitted, they labeled the DNA in the phage with radioactive isotope of phosphorus 32 and the protein code with the different radioactive isotope of the sulfur 35. They then allowed the labeled phage to infect the bacteria and examine which radioactive marker ended up inside the bacterial cell. Here the life cycle of the phage. It entered the bacteria and breakdown of the bacterial chromosome totally broken down, and the phage chromosome start to replicate, and the expression of phage genes to produce structural component, assembly of the progeny, and the release of progeny phage, and the cycle. Here the bacteriophage is composed of DNA and protein. Set up to replicate, label DNA with phosphorus 32 and label protein with sulfur 35. Infect E. coli bacteria with two types of label T2. B32 is discovered within the bacteria and the progeny phage, whereas S35 is not found within the bacteria but released with the phage ghost. Here, shrimp tobacco mosaic virus experiment in 1953. The viral strain to demonstrate RNA is the genetic material of tobacco mosaic virus. RNA also is a genetic material in some viruses, and this is Buzaj experiment. Gare and Schramm in 1956. He is a German. Experiment on tobacco mosaic virus demonstrated that the viral RNA alone is sufficient to induce viral infection in host cell. They extract and purified the RNA component from tobacco mosaic virus and found that it was still able to infect and replicate within the host cell. This was a pivotal experiment that helped establish RNA as a genetic material. And he gave a lecture in Max Planck Institute, explain his experiment. For conclude these experiments, the early experiment, Griffith and Avery, DNA not RNA is transforming agent. Hershey and Chase, DNA not protein is the genetic material. Gare and Sherm, RNA not protein is genetic material for some viruses. The nucleotide, here the structure of DNA, 3 component, the pentose sugar, 5 carbon sugar, DNA desoxyribose and RNA ribose, compare the 2 carbon, and nitrogenous base, purine, adenine, guanine. This is the nitrogenous bases. Nitrogenous bases is 2 types, purine and pyrimidine. Purine, adenine, and guanine. Pyrimidine is cytosine, thymine, and uracil. Thymine in DNA and uracil in RNA. This is the structure of DNA, as we know. The ribose, the sugar, the 5 carbon sugar and the nitrogenous bases. Phosphate group attached to the phosphate group attached to the 5 carbon. Nucleotides are linked by phosphodiester bond to form polynucleotide. Phosphodiester bond, covalent bond between the phosphate group attached to 5 carbon of the nucleotide and the 3 carbon of the sugar of another nucleotide. This bond is very strong, and for this reason DNA is remarkably stable. DNA can be boiled, even autoclaved, without degradation. The ends of the DNA or RNA chain are not the same. One end of the chain has 5 carbon and the other has 3 carbon. Here the 5 end are here the 3 end. Watson and Crick model the double helix. Double helix model of DNA, 2 sources of information. Base composition studies of Erwin Shergaff indicated double-strand DNA consists of 50% purine and 50% pyrimidine, and this amount of amount of T, Shergaff rules, and the amount of G equal to the amount of C. C percentage content varies from organism to organism. Here the Homo sapiens, the human being, A and T and G and C and G, C, this is the percentage of G and C and T. The mice and Drosophila, here we find Watson and Crick explain their model. The James Watson and Francis Crick in 1956 by the X-ray diffraction studies done by Rosalind Franklin and Merrick Wickens, conclusion DNA's helical structure. Here the picture taken by X-ray crystallography by Rosalind Franklin, and this is the first picture for the DNA. A bird biologist and expert on structure of charcoal, designer of underwater mine, a nuclear physicist. Those are the heroes of the story. The result, a nuclear physicist intersected, resulting not in the submarine explosion of feathers as one might expect, but in discovery that offered a glimpse of molecular mechanism that underlie all life. In the insight, innovation, and persistence of James Watson, Rosalind Franklin, Francis Crick, and Merrick Wickens led to detailed understanding of the structure of DNA, the stuff that genes are made of. There's all their knowledge integrated to discover the helical structure of the DNA. Double helix model of DNA, 6 main features. 2 polynucleotide chain wound in a right-handed clockwise double helix. Nucleotide chain are anti-parallel. 5 phase 3, 3 phase 5. Sugar phosphate backbone are on the outside of the double helix, and the bases are oriented toward the central axis. Complementary base pairs from opposite strand are bound together by weak hydrogen bonds. The pair with 2H bond and she pairs with the 3H bond. Here, base pair are 0.34 nanometer apart. One complete turn of helix require 3.4 nanometer, 10 bases per turn. Sugar phosphate backbone are not equally spaced, resulting in major and minor grooves. Adenine thymine base double hydrogen bonds. Guanine cytosine base triple hydrogen bonds. Guanine cytosine triple hydrogen bonds. Thymine adenine 2 hydrogen bonds. Here we find the double helix DNA. This is the backbone of the double helix, and thus Nobel Prize in Physiology and Medicine, Maurice Buchan and Francis Crick and James Watson. Rosalind Franklin has a picture of the DNA by crystallography. RNA appear with U and C pairs with G. Messenger RNA, there is transfer RNA, ribosomal RNA, small nuclear RNA, all are single-stranded function in transcription, RNA processing and translation. Here we find the messenger RNA, its structure. The organization of DNA, RNA in chromosomes. The genome is composed of chromosome or set of chromosomes that contains all the DNA an organism or organelles possesses. The viral chromosome, single or double-stranded DNA or RNA, circular or linear, surrounded by protein. This is the mosaic virus, tobacco mosaic virus. This is the tutu bacteriophage. This is the lambda bacteriophage. Most contain one double-stranded circular DNA chromosome. Other consist of one or more chromosome and are either circular or linear, typically arranged in a dense clump in a region called the nucleoid. The problem measure linear. The Escherichia coli genome 4.6 MB would be 1000 times longer than the E. coli itself. How this genetic material find a space inside the cell? The human genome is 3.5 GB, would be 2 to 3 meter long if stretched linearly. What is the solution? How the genetic material find a space inside the cell? The solutions are: number one, supercoiling. DNA double helix is twisted in space about its own axis, a process is controlled by topoisomerase enzymes, occur in circular and linear DNA molecule. Looped domain. More about genome. Here, a loop are attached at the base is unknown way. More about genome size. C value, total amount of DNA in the haploid 1N genome varied widely from species to species and show no relation to structure or organizational complex. Eukaryotic chromosome structure. Chromatin, complex of DNA and chromosomal protein. Twist as much protein as DNA. Two major types of protein, histone and non-histone. Histones, abundant basic protein with a positive charge that bind to DNA. 5 main types, H1, H2A, H2B, H3, H4, equal in mass to DNA, evolutionarily conserved, non-histone. All the other protein associated with DNA differ markedly in type and structure. Backing of DNA inside the chromosome into the chromosomes. Level 1, winding of DNA around the histone to create a nucleosome structure. Level 2, nucleosome connected by strand of linker DNA like beads on a string. Backing of nucleosome into 30 nanometer chromatin fiber. Formation of looped domain. Chromosome DNA is packed inside the macroscopic nuclei with the help of histone. These are positively charged proteins that strongly adhere to negatively charged DNA. And this is the level complex called nucleosome. Each nucleosome is composed of DNA around 1.65 times around each histone protein. This is the level DNA double helix. Beads on a string. Form of chromatin. Chromatin fiber of packed nucleosome. Extended section of chromosome and condensed section of chromosomes. Here some terms about DNA, centromeric DNA, center of the chromosome specialized sequence function with the macrotubule and spindle apparatus during mitosis and meiosis. We need it in mitosis and meiosis. Telomeric DNA at extreme ends of the chromosome, main stability. It's responsible for the main stability and consists of tandem repeats, play a role in DNA replication and stability, and the aging of the cell. Unique sequence DNA, often referred to as a single copy and usually code for genes. Repetitive sequence DNA may be interspersed or clustered or clustered and vary in size.